Okay, you guys. This part right here is just for sixth hour. The rest of you have already written this down, and so you can fast forward a little bit until I start with chapter 45 stuff. But for sixth hour, what we need is I need you on the back of your chapter 11 notes. There's a blank sheet of paper, and you need to write this information about this process called apoptosis. And so you can pause this and write it from the screen, and I'm going to go ahead and talk about it. So it's programmed cell death or cell suicide. So how this relates to cell signaling is that the cell can release what's called a death ligand and cause it to die. How does it do that? So what's activated is the enzyme, and what happens is the enzymes chop up the components of the cell. So that means organelles and things like that, and then they package them into vesicles that are later digested by scavenger cells. Scavenger cells are cells that can undergo endocytosis to take in particles and digest them. And so what happens is this process of the interior part of the cell being digested. So we have this cell, and there's a death ligand that binds to it. So that death ligand can be produced by that same cell and goes and binds to the receptor. And what that triggers is a signal transduction pathway that eventually causes an enzyme or a groups of enzymes to be made and activated, and then that triggers the destruction of the cell. And so therefore, you don't want all these digestive enzymes to get out of the cell and start digesting surrounding cells. So what happens to this cell here is eventually it packages all this digestion material and causes, and the cell becomes, instead of one big cell, it breaks off and becomes many smaller vesicles. And the vesicles contain parts of the cell, and then these vesicles are the vesicles that are digested by scavenger cells. And so that's what happens. So this apoptosis, the cell death, can be triggered by that extracellular outside the cell death signaling ligand, which that's an example of up here. Also, if DNA is damaged, so if you have damage to the DNA and you don't want that cell then that is damaged, because the DNA codes for making proteins, so if it's damaged, you don't want to make more cells of that, and so therefore the cell can be triggered to die. Or if the cell, the DNA is okay, but then it's folding the proteins wrong in the endoplasmic reticulum, and it's not getting its right shape, then those proteins aren't going to have the right job and be able to do the right job in the cell, and therefore there's going to be something going on that's wrong with the cell, and so therefore that can also trigger cell death. I'm going to flip this sheet over because there's 3 more bullets that you need to write. So again, you might want to pause this. So apoptosis, why it's important, as we said here, can kill off cells that have damage done to them, so that's a good thing. It's also part of normal development in animals, and so an example is between our fingers. So here's my hand here. Early on in development, there's tissue between our fingers, and this tissue slowly goes away during development, and why it goes away is the cells that comprise that tissue goes through apoptosis, and so these cells die, and therefore leaving our digits. And this happens with our hands and with our feet. And on Monday, I'll show you a picture of that happening with the paw of a mouse as well. Apoptosis has also been linked to some things that are harmful. So it's thought that apoptosis may be involved in some diseases like Parkinson's and Alzheimer's, where apoptosis is going on in cells that maybe shouldn't be going on. And then in this case here with cancers, they think that cancers bypass apoptosis, so these cancerous cells, which there's something wrong with them, and cancerous cells, what they do is they divide uncontrollably. So normally whether when a cell divides and becomes two cells, that's tightly controlled. And if something is wrong with that regulation system, that's something wrong with the cell, which should trigger apoptosis and doesn't. And so therefore you can have a cell with something wrong with it and not regulating its division, cell division, and reproducing so that you can get lots of those cells that have something wrong with them. And so we'll talk a little bit more about cancers in chapter 12. All right, so that's for 6th hour, and so that's what we missed at the very end of the hour. So now we're going to start with the chapter 45. So everybody else, this is where we're going to start here. And the first part of this chapter is really, as you'll see, quite a review of some of the things that we've already talked about here. So notice it says hormones in the endocrine system. So the endocrine and nervous system. We're going to talk about the nervous system in the coming weeks, and nervous and immune system. So I'm going to introduce it to you now. So these two systems coordinate communication throughout the body. The nervous system sends certain signals and the endocrine sends certain signals, but the signals are different. So it allows the parts of your body to communicate. So the endocrine system secretes hormones that travel via the blood and coordinates slower but longer acting responses, including reproduction, development, energy, metabolism, growth, and behavior. The endocrine system often sends hormones, like we said, through the bloodstream, and the blood travels throughout your body. And so it takes a little time for those hormones to get what they're supposed to get and bind with the cells that they're supposed to bind with the correct receptors, and then get the response going. So it's a slower process than what happens in the nervous system. The nervous system conveys signals as well, but they convey high-speed electrical signals. So these are much different signals. The similarity is they're both sending signals. The electrical signals travel at a much faster rate. So, and they travel through these specialized cells called neurons. The neurons is the basic cell for the nervous system. And what happens is these cells regulate other cells. So what that means is like a neuron can stimulate a muscle to contract. So therefore a muscle doesn't contract unless a neuron tells it to. And so that's what it means by controls other or regulates other cells. The nervous system transmits information between specific locations, so like the spinal cord and a muscle cell. Nerve impulses can be received by neurons. They can be received by muscle cells, endocrine cells, and exocrine cells. So these neurons, so you have one neuron. A neuron kind of looks like this. It has a cell body, but coming from that cell body, it has these little fibers called dendrites, and then it has this long part here called the axon. All right, and then it has this synaptic terminal at the end here. And so what happens is that this neuron communicates with another neuron by releasing chemicals called neurotransmitters. So these guys leave the neuron. So this is one neuron. And so what we're talking about is that the receiving cell, who's going to get the neurotransmitters and therefore cause a response to happen, the receiving cell here could be a neuron, a muscle cell, an endocrine cell, or an exocrine cell. And so that's what these guys here is, the receiving cell from the neuron. So what are exocrine and endocrine glands? Exocrine glands are glands that secrete their products, excluding hormones and other chemical messengers, into ducts, which lead directly to the external environment. So ducts are like little tunnels. So example, sweat glands. Sweat glands, you have cells of, this is the surface of your skin, you have cells that secrete your components of sweat into these glands, and then there's a duct that then leads to the external environment, which in this case is the surface of your skin. So that's what that means there. Mammary glands, so in the breast tissue of mammals, there are glands that cells that produce milk, and then they go into the glands and through the ducts and then leave the body and can go into the mouths of babies. Salivary glands in your mouth. Your stomach, pancreas, liver have glands that produce certain enzymes. They have ducts that allow them to mix the enzymes to mix with your food. They are the counterparts to endocrine glands. So endo is the opposite. Exo is outside, endo inside. So these guys secrete their products, which happen to be hormones, directly into the bloodstream. So these guys are ductless glands. There are no ducts, then there's nothing that tunnel it travels through, like I used the example with the sweat. Or they release hormones that affect only target cells nearby. So the cells can be nearby, where it releases a ligand, and then that ligand can bind to a nearby cell. Or the cells can be far away, where here's your bloodstream. The ligand is produced by one cell, the hormone, and then it goes to another cell in another part of the body. It can either go in or, depending upon if it's polar or nonpolar, binding to the receptors or into the cell. All right, so let's look at endocrine signaling and focus on this. This is going to be the focus of this chapter, or what we're doing for this with this chapter. So hormones secreted into the extracellular fluids by endocrine glands reach their targets via the bloodstream. So that's like this here. So that it releases first into the extracellular fluid, then into the bloodstream, which then can go to the target cell. Hormones may travel long distances. All right, so it can go all throughout the body and travel very long distances. Endocrine signaling maintains homeostasis, which remember is maintaining a constant internal environment. So that can be regulating lots of different things, body temperature, and so on. So lots of different things. We're going to look at in a little bit here about maintaining glucose levels. It can also mediate responses to stimuli. So that's external things, and also regulate growth and development. So these hormones do lots of different things. So as a review, we have part of the endocrine signaling. We have paracrine and autocrine signaling. And so local regulators act over short distances, reaching target cells solely by diffusing. And so in paracrine, if you remember from our cell communication packet, paracrine signaling, the target cells lie near the secreting cells, so they're close. All right, so that's like this example up here. They're close together. In autocrine, the target cell is also the secreting cell, so it is communicating with itself. So here it produces the ligand and then communicates with itself, autocrine. And those are both types of endocrine signaling. So this picture here is showing you those different things. So endocrine signaling is long distance, a type of cell communication, and so the one cell makes the ligand, it goes into the blood vessels, travels long distances, and goes to the target cells. Notice here these little spots on the membrane of the target cells are supposed to be the receptors. So these cells have the right receptors. See these little receptors here. So these ones have the right receptors, and therefore inside then that elicits a response. So a signal transduction pathway which gives you a response. So that's endocrine system signaling, long distance signaling there. Oftentimes going through long distances and oftentimes using the bloodstream. So that's one type of signaling. Paracrine signaling is the cells are nearby, so this cell is going to communicate with these cells, so they're nearby, so you don't really need the bloodstream. It can just diffuse through the extracellular fluid, and the cells that have the correct receptor, so these cells right here do not have the correct receptor, so they're not being affected by these ligands. These guys are, and so therefore they elicit a response. And then here's the autocrine signaling, where it releases the signal and the signal binds to receptors on the same cell. So hormones, remember, can be in two classes, non-steroid or steroid. So the non-steroid hormones do not pass easily through cell membranes. They're often amino-based, so amino acid-based, which means that they are proteins, all right, or polypeptide chains. Most hormones are like this. So most of our hormones, we're going to talk about insulin. We've already talked a little bit about epinephrine. I'm going to go over it again real quick. And then our second class of hormones are steroid hormones that do pass easily. Testosterone is an example. Testosterone, sorry. And estrogen are examples of that. So they don't pass through the they do pass through the cell membranes. They don't need a receptor as part of the membrane. So remember, these guys go into the cell, and that's where the receptor will be. These guys, the receptor is part of the cell membrane, and they bind to it. So water-soluble hormones. So let's talk about insulin, epinephrine. They bind to a receptor. So the binding of a hormone to its receptor initiates that signal transduction pathway. And this leads to a response in the cytoplasm or enzyme activation. So it activates an enzyme that has already been made, or it can have a change in gene expression. So it can activate, I'm going to put here, activate a transcription factor which can turn on a gene. And therefore can make a protein that it never was able to make before. So let's go through an example of a water-soluble hormone that we've already talked about, epinephrine. It's multiple effects on the body's response to short-term stress. And so this is like the fight or flight response, where the body, you're in a stressful situation and you need to very quickly either fight the situation or flee, or flight means to run away. And so either way, you need some energy. How do you get energy? You need glucose to go through cellular respiration to make ATP. All right, so what happens is in this short-term stressful situation, epinephrine is released. This is a hormone. What it does is binds to receptors on different cells and on the plasma membrane of, and I'm going to use the example of liver cells. But it has multiple effects. We talked yesterday about a different effect that it has, epinephrine has on liver and heart cells. So therefore it has some different varying effects. So when it binds to the liver cells, what the response is this triggers the release of messenger molecules that activate enzymes. So on the liver cell, epinephrine binds, goes through a signal transduction pathway, activating the enzyme that's going to digest and break down glycogen. And so what happens is it activates enzymes and results ultimately in the release of glucose into the bloodstream, which I'm going to put in parentheses here, can be used in CR, which is cellular respiration, for ATP production. So therefore, what does this mean? You get this glucose released and you can get energy. And why do you need energy? Because you're in this short-term stressful situation, and so therefore need some glucose for energy. So that's how that works. And so this next picture, this just builds on it. This is the whole picture here, so I'm going to focus on this. This is figure 45.7. in our textbook. So here, relating to chapter 11, here's your G protein linked receptor. Here's your epinephrine binding to it. This changes shape, which then activates the G protein, which then activates the enzyme adenylyl cyclase. What does this enzyme do? It catalyzes the reaction of converting ATP to cyclic AMP. So now you have cyclic AMP as your second messenger, and that activates. So this is made, this activates, second messenger activates the protein kinase A, which then goes through a signal phosphorylation cascade and eventually has two responses. The promotion of glycogen breakdown. So what is glycogen? Remember, it's a chain of glucose molecules. Connected there, connected, dot, 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 dot. All these glucose molecules. So when it breaks down, you just have glucose. And this glucose, they're not connected together, can be used for cellular respiration for energy. And so that's your response, the glycogen breakdown. Where does glycogen stored? In your liver cells. So that's where it's happening. So back over here, when we said that epinephrine is going to bind to receptors on the liver cell, all right, this right here is your liver cell. And so here's epinephrine binding to it. So this is the membrane of it, and this is inside of it. So you get glucose, and then where does the glucose go? It's gonna be transported out of the liver cell and into the bloodstream. So out of the liver cell, here's the liver cell, here's the rest of this. The membrane here. So it goes into the blood and it goes to the different cells of the body to be used for energy. All right, so then remember that hormones that are not polar can get through this lipid bilayer. So these guys, a lipid-soluble hormone or a nonpolar or hydrophobic hormone is usually the change it doesn't bind here, and so the change or the response in the cell is different, and this actually regulates oftentimes gene expression. So therefore the hormone enters target cells and binds to protein receptors in the cytoplasm or actually inside the nucleus. And then therefore these proteins receptor complexes, once the molecule binds to them, regulates the transcription of specific genes. So what does this mean? So this is your cell, here's your nucleus, here's the receptor. And so what happens is that your molecule can go into the cell and bind. I should draw the molecule so it shows the specific shape here. So this molecule, the receptor, can either be in the cytoplasm or in the nucleus. I'm drawing it in the cytoplasm here, but it could also be actually in the nucleus, which triggers a signal transduction pathway. The molecule at the end can activate a transcription factor. So the transcription factor right here, it becomes activated. So it activates transcription factor. It binds to a gene. So here's the gene. And what that does is this is the gene. And transcription means that you're going to take the gene in the DNA and make an mRNA molecule. That mRNA leaves the nucleus and goes to the ribosome. So this is the ribosome, and that's where your protein is made. And so that's kind of what it does. So it turns on the gene. So when it says regulates the transcription of specific genes, that's how it does that. So that's a quick little review of that. And so that's what this picture here is showing you. Again, this is the beginning part of it, and this is building on the same picture. So I'm going to just look at this bottom picture. So you have a hormone that enters the cell. It is lipid soluble, and so it can get through the lipid bilayer. So it comes in, here's its receptor, there's the hormone receptor complex, it enters the nucleus. Sometimes that can directly act or bind to the gene. So I was trying to relate last chapter here. Sometimes it can activate a transcription factor that binds to the gene, and sometimes the actual complex with the hormone bound to it can actually bind to the gene. So then from the gene, so here's the, let's say here's the gene, you make an mRNA from that gene. So this is a gene, and then that mRNA goes to these guys here, which are your ribosomes, and make sure this is your, right here, your polypeptide. So that's what it means by regulating transcription. So this part, the making of the mRNA, would never happen without your hormone. And so then you never get this particular polypeptide. Okay, so as we talked about, hormones can have multiple effects. So the same hormone may have different effects on target cells that have different receptors or different signal transduction pathways. All right, so this picture shows you that in a little bit different way than we talked about in chapter 11. So notice here we're comparing two different things. So it says same receptors but different intracellular proteins. They're not showing you the intracellular proteins, just telling you that they have different proteins. So, and then that makes them have, because they have different intracellular proteins, like the signal transduction pathway proteins, they're going to have different cellular responses. So notice here there's this bracket, so we're going to compare these two cells. What kinds of cells are these? Liver cells and skeletal muscle cells. And so if we look here, these two cells are the liver cells and the skeletal muscle surrounding a blood vessel. So there are muscles surrounding blood vessels, and so and it helps the vessel to constrict, which means the diameter gets smaller. So from like this to smaller like this, or dilates, which means the diameter gets bigger. And so, so what happens here is, notice that here's the B receptor. They call a beta receptor, and there's epinephrine, and so it's the same ligand, same receptor, they bind to it. But what happens inside is that triggers this, since they have different intracellular proteins, you have a different signal transduction pathway, which leads to a different response. And so inside here has a different signal transduction pathway, which leads to a different response. So what is the response? So here, the response is the glycogen breaks down, this is in the liver cell, and glucose is released from the cell. So that's just like we just talked about just a little bit ago about what epinephrine does in the liver cell. So this is the response here, and that's a different response than here. So here the response, even though we have the same receptor, the vessel dilates. So this is the response here. Okay, so that's same receptors, different proteins inside. Next thing is different receptors, which then lead to different cellular responses. So here we have, again, epinephrine is the same. We're looking at now the skeletal muscle around the blood vessel and your intestinal blood vessels. So what does that mean? Blood vessels around the intestines, small intestine, and so on. So they have different receptors here. Notice here it says alpha receptor. So they have a slightly different shape, but what's similar about the shape? The active site there, the spot that the epinephrine is going to bind to. But the receptors are different, which then triggers ultimately different responses. And so here we have, this is the response, is in this case here, the vessel constricts. And so therefore we have a constriction there. And so just showing you the two different things, different intracellular receptors and different receptors. All right, so looking at the pathway of hormones, simple hormone pathways. Pathways are released from an endocrine cell, travel through the blood, or bloodstream, and interact with specific receptors within a target cell to cause a physiological response. So I'm going to give you an example. For example, the release of acidic contents from the stomach into this right here is the beginning of the small intestine. which then stimulates the endocrine cells there to secrete the hormone secretin. This causes the target cells in the pancreas, which is a gland that's kind of located behind the stomach, and what that's going to do is raise the pH in the small intestine. So let's give an example of, just to give you a picture. Here's the esophagus from where you eat your food, and then you swallow it. It goes down your esophagus, which leads to your stomach. So here's your stomach. And then your stomach is connected to your small intestine. And so where is the pancreas? So then the pancreas is back over here. Let me pick another color here. Pancreas is I'm going to kind of show it sticking out here, but the pancreas has a small tunnel, a duct, that leads from the pancreas to the beginning of the small intestine. All right, so that's how everything's kind of connected. And so if we look here, remember that your stomach, the cells represented by this pink line, the cells of your stomach release acid. And so when it releases acid, remember there's other cells that release enzymes, and the enzymes in your stomach need an acidic environment to work. So there's the purpose of the acid. But it doesn't destroy the cells of your stomach, because you also have cells that release mucus, and that mucus protects the lining of your stomach. So your lining of your stomach isn't harmed by the acid, but what that does is lower the pH. And you have sphincter muscles. There's a muscle here. Sphincter muscle is a circular muscle that can open and close, all right? And so when your acidic contents don't go back up into your esophagus. And so anyway, so it lowers the pH of your stomach. And so then what happens is some of your food begins to digest, and then it goes into your small intestine. Now your small intestine, now you've got this acidic contents in your small intestine. Your small intestine does not have the cells to make mucus, so they don't have that protection. So it can be detrimental for the cells lining your small intestine to have this acidic nature. And so then what happens is, to neutralize and bring the pH back up so it's not so acidic, your pancreas actually releases what are called bicarbonate ions. So I'm going to write here, they write bicarbonate, all right, ions, and it releases them, and it goes down the duct and mixes the bicarbonate ions, the bicarbonate fluid mixes with your food and neutralizes it. All right, and so that's kind of the big picture of what's going on here. So now what I want to talk about is how cell communication and how hormones play a role in, so it actually plays a role in the pancreas knowing when to release the bicarbonate ions, because the pancreas only needs to release it when you have acidic digested food coming into your small intestine. So that's what the next picture shows you here. So this is the relatedness, or how this is related to cell signaling. So your stimulus. So here your stimulus is to cause the pancreas to release the bicarbonate ion is a low pH. Remember, this is the beginning of the small intestine. So back in my picture over here, you have food entering in, the stomach releasing the acid, and now you've got this acidic digested food moving into your small intestine. So your small intestine now is getting a low pH, all right, because it's more acidic. So we want to fix that. And so that's the stimulus. We have low pH in the small intestine. So then notice here it says the S cell. So these are just a type of cell in the small intestine. secretes the hormone secretin. All right, so that's represented in this picture by these little dots. So this right here is your small intestine cell. It's what it says, endocrine cell. That's what it is, small intestine. It's secreting, this is the hormone secretin. How does the hormone, remember what we want is we want this hormone that's being secreted by the small intestine to communicate with the cells of the pancreas. And so that means the pancreas cells has to have receptors for it and so on. So the small intestine releases or makes secretin and puts it into the bloodstream. And so the bloodstream, I don't have a red pen here, but let me try and draw the add on to this. So there's the bloodstream that passes by the small intestine, but also surrounds the pancreas. So this is your blood. And so this molecule called secretin, so this is secretin, enters the blood, and that this is made, so this is secretin. This is made in response to the low pH. It enters the bloodstream and travels and then goes to the cells of the pancreas. So how does it do that? It's going to bind to a receptor on the cells of the pancreas. So the pancreas has the right shaped receptors. So the hormone secretin goes now into the bloodstream, which then is carried to your the target cell, which is your the pancreas. So this is your target cell. And then, so what does that mean? So here on the target cell, the pancreas cell, there are receptors. They have the right shape. The secretin can bind to it. That triggers a signal transduction pathway that elicits a response. What is the response? The response is the release of this bicarbonate that's going to bring the pH up. So it was, remember, it was the pH was too acidic. And so this bicarbonate ion, so the secretin goes into the bloodstream, communicates with the pancreas. The pancreas now makes bicarbonate ion and secretes it through this duct now to the small intestine, and the small intestine is going to decrease in pH because the bicarbonate ion will bring it, increase the pH, bring it and make it less acidic and more neutral, which is what it should be. All right, it actually makes it a little bit basic, actually. All right, so that's how that works. And so now notice here that your end response, eventually when the pH is brought back up, you want to stop this from happening. So you don't want to keep on bringing the pH up and up and up till it gets very, very basic, like a pH of 14. So therefore, when the pH is brought back up, that response actually will inhibit your initial stimulus because now you won't have a low pH in the small intestine. And so then that inhibits this process here. That's called negative feedback. So that's what this next little bit is. Feedback regulation. A negative feedback loop inhibits a response by reducing the initial stimulus, thus preventing excessive pathway activity. So that's what this is showing you here. So it reduces once you get the response, it reduces the initial stimulus, which means you're going to have less of that response until, of course, you have, in this case here, new food coming into the small intestine that lowers the pH again, and that'll stimulate it again, and so on and so forth. And so that's a negative feedback loop. This is what one of your packets for homework is about. When it says feedback mechanisms, negative feedback is one of those. Another type of feedback loop is positive feedback. Positive feedback reinforces a stimulus to produce an even greater response. And so when would you want the response, you want the stimulus to keep going and to get a greater response? So an example is in mammals, oxytocin, this is a hormone, causes the release of milk. So in the breast tissue, in the milk, this causes, the more milk that's produced causes greater suckling by the offspring, which stimulates the release of more oxytocin, And the more oxytocin then causes more milk to be released, which then and then stimulates the release of more oxytocin and so on. So therefore, as one of these occurs, it causes the next one to occur even greater, and so it stimulates it. And so that's positive feedback. Negative feedback, the response inhibits or stops the stimulus, and so this actually encourages it and causes it to happen more. And so that's what your feedback mechanism packet is about. It gives some examples of positive and negative feedback. And then the second packet is specifically about two different hormones that control blood glucose levels in our blood. So these are two hormones that I want you to know about. They are insulin and glucagon. So insulin decreases blood glucose levels, and glucagon increases blood glucose. And they are antagonistic hormones, which means that they have opposite effects. So the gland that makes these and releases these is the pancreas. The pancreas has clusters of endocrine cells called pancreatic islets. There's two different kinds. There's alpha cells that produce glucagon, and there's beta cells that produce insulin. So let's look at the next picture to see how these two hormones regulate blood glucose levels. So here we have in the center, here's our homeostasis. This is our blood glucose level, and this is normal. So this is the concentration that's normal, between 70 and 110 milligrams. So that's it's a range. So it's normal for some people may be 80, and normal for another person might be 90. It's a range, but in that rough range is where we want to keep it. So what happens is sometimes our blood glucose levels get higher than that range, and sometimes they get lower. And so what happens is our pancreas helps to control that. So let's look at the first situation. So notice here this arrow here. So the stimulus is what happens if your blood glucose levels rise. So when would a blood glucose levels rise? This is after you eat a carbohydrate-rich meal. So you just ate a bunch of pasta. Pasta is made out of starch. Starch is chains of glucose. When you digest it, you break down the chains of glucose into individual glucose molecules, which enter your bloodstream, and now you've got what we call high blood sugar levels. So that's what it means. So in layman's terms, some people might say or talk about high blood sugar levels. So what does that do? That's the stimulus. So what does that stimulate? That stimulates the beta cells of the pancreas to release insulin into the blood. So insulin is now in your bloodstream and can go to all the cells of your body. And the thing about insulin is that it's needed for cells to take in glucose. So without insulin, a cell can have glucose on the outside and can't get into it. The glucose can't get into it. And so that's the importance of this hormone, insulin. So it goes into the bloodstream, so it's now in the blood, getting carried to all the body cells. And when it binds to your body cells, it helps your body cells to take up more glucose. So therefore, that's taking glucose out of the blood, and so therefore the blood glucose levels are going to start to decline and go back to normal. The second pathway, so that's the first thing, is your body cells. The second thing is that your liver cells start to take up glucose. And remember, it stores it as glycogen. So it cooks the glucose together. And so this is your storage of glucose until you need it for later use. And so both of these processes take glucose out of the blood, and therefore the blood glucose levels decline, and you go back to your homeostasis level. So insulin's very important. On the flip side, what happens if your blood glucose levels goes below normal? So in this case, here's the stimulus is blood glucose level falls. So they have low sugar levels in your blood. For instance, after skipping a meal. So you don't have as much sugar floating in your blood. So therefore, what your body does is try to bring that sugar level up in your blood. And how does it do that? Well, first of all, that stimulus stimulates your alpha cells of the pancreas to release glucagon. So this is another hormone into the blood. And so glucagon is now in the blood and can go to all the different cells of your body. And where it goes is the place where you store your glucose, which is in the liver. So this glucagon stimulates your liver to break down glycogen. So remember, glycogen, when we break it down, it was chains of glucoses. And then what happens is we break these bonds between the glucose levels, or glucose molecules, and now you have individual glucose molecules that can now go into your blood, and therefore your blood glucose levels rise, and it brings your blood glucose levels back to normal. And so this kind of figure 8 here keeps on happening to keep your blood glucose levels where they're supposed to be. So those are antagonistic hormones. So glucagon, what are you going to write? Increases or decreases blood glucose levels? You should have it increases. And how does it do that? By colon. There's two different ways. Stimulating conversion of blank to glucose in the liver. So you might want to pause this and see if you can fill in the blanks of these two here. Just play that little game here, see if you can do that, fill in all the blanks of just these two bullets here. So pause that right now. I'm going to continue to go on. Stimulating conversion of, and you should have, hopefully you got glycogen to glucose in the liver. You probably had trouble with this one, stimulating the breakdown of blank and blank into glucose. Remember when we talked about cellular respiration, we can actually convert fat and proteins into glucose, and they can enter into the cellular respiration process. Insulin also, then, you should have reduces or lowers the blood glucose levels by, it can promote, what can it promote? The cellular uptake of glucose. So it can take, so glucose can start to go into the cells. It slowing glycogen breakdown in the liver. So glycogen breakdown doesn't happen. So you're not defeating your purpose of having the body cells over here, having the body cells take up the glucose. And then it promotes fat storage, not the breakdown. So you have too much glucose. Some of that glucose will be converted to fat, and you'll store it. All right, and so that is our two hormones, and I just want to spend just a minute and talk about when this system that we just talked about is disrupted. And so we have a couple of different kind of disease, or one disease that I want you to know, diabetes mellitus, or sometimes just called diabetes. It's probably the best known endocrine disorder. It's caused by a deficiency of insulin or a decreased response to insulin. So remember, what does insulin do? Insulin over here helps the cells of your body to take up glucose and bring your sugar levels back to normal. And so in this case here, if you don't have insulin or your insulin's not working up to par, you're going to have high blood sugar levels because remember, insulin is needed to get the glucose into your cells, so that means the glucose will stay in your blood, and you'll have high blood sugar levels, which means then your cells aren't getting enough glucose. All right, is basically what's happening. So it's marked by elevated blood glucose levels. So there's two different kinds of diabetes, type 1 and type 2. Type 1 is insulin dependent. Sometimes they call it juvenile diabetes because oftentimes it hits individuals when they're young. And so type 1 diabetes is insulin dependent. It's actually an autoimmune disorder. In which the immune system destroys the pancreatic beta cells. And so if we go back over here, so autoimmune disorder, auto means self. And so what happens in any kind of autoimmune disorder is that your own immune system is supposed to attack and kill foreign cells or foreign particles that are coming into your body. When something goes awry and your immune system attacks its own cells, that's called an autoimmune disorder. And so what happens is people with type 1 diabetes, something triggered their immune system to attack and destroy the beta cells of the pancreas. So going back up here, here's your beta cells of your pancreas. And they're supposed to release insulin so that you can control your blood sugar levels and take glucose out of the blood and your cells can use it. So if in type 1, these beta cells are all completely destroyed, then they don't make any insulin, and therefore their cells of their body can never get glucose, which means that they'll starve. And so therefore people with type 1 diabetes are insulin dependent, which means that they have to take shots of insulin every single time that they eat to mimic what their cells would do, so that when you eat and you get your glucose in your blood, you manually give yourself insulin so that your body cells can take up the glucose and use it for cellular respiration. So that's type 1. Type 2 is often non-insulin dependent. Sometimes eventually maybe the people have to eventually take insulin, but most people that's not the case. So what happens is it involves insulin deficiency, which means they don't have enough insulin, so they have reduced responses to in the target cells due to a change in the insulin receptors. So in the insulin receptor in the cell here, this insulin is supposed to bind to it. If you have a change in the receptors, then it doesn't matter if it's hard to get the insulin to work because the receptors have changed. In type 2 diabetes, like I said, most people don't have to take insulin shots. Sometimes there's some medications that they can be used to help to regulate the blood glucose levels. Type 2 diabetes, there's a strong genetic component to that. It runs in families. Also, there's a strong environmental component, meaning that your diet and your health can play a role. So type 2 diabetes can be triggered by being overweight, and so on. So oftentimes, if that's the case, it can actually be reversed. So somebody who is overweight can begin to lose weight and eat healthy, and so on, and reverse the effects and become not need their medication and not have diabetes anymore. So they're very different forms of diabetes, but they both have the same kind of end result as a result of that. And that, my friends, is the end. So it's not even an hour long, 52 minutes. So I hope you have a great Thanksgiving, and I'll see you on Monday.